Good morning, students. This is Dr. Divya. Today's topic is History of Biotechnology. What is biotechnology? Biotechnology is the branch of molecular biology that studies the use of microorganisms to perform specific industrial processes. Biotechnology produces genetically altered bacteria that solve the problems. So biotechnology in before Christ, 500 before Christ, like this Chinese use moldy curds as a antibiotic to treat boils. 250 BC, the Greeks practiced crop rotation to increase soil fertility. 100 BC, Chinese use powdered chrysanthemum as an insecticide. Pre-20th centuries biotechnology. 1590, Janssen invents the microscope. 1663, Hooke discovers cells. 1675, Leeuwenhoek discovers bacteria and protozoa. 1797, Jennifer included a child with a viral vaccine to protect him from smallpox. 1802, first time the term biology is used. 1830, proteins, the building blocks of cells, are discovered. 1833, the nucleus of the cell is discovered. 1855, the E. coli bacterium is discovered. 1855, Pasteur works with yeast, eventually proving they are living organisms. 1863, Mendel discovers genes while working with bees. He lays the groundwork for genetics. 1879, Fleming discovers chromatins. 1883, the rabies vaccine is developed. 1888, Waldeyer discovered the chromosome. 1902, the term immunology first used. 1906, the term genetics is used. 1915, bacterial viruses called phage are discovered. 1919, the word biotechnology is first used. 1927, Muller discovers that X-ray cause mutation. 1928, Fleming discovered penicillin. 1938, the term molecular biology is used. 1941, the term genetic engineering is first used. 1942, the electron microscope is used and characterizes the viruses that infect bacteria called bacteriophages. 1944, DNA is shown to be the building block of the gene. 1949, Pauling proves that sickle cell anemia is a molecular disease caused by a mutation. 1953, Watson and Crick understand the structure of DNA. 1954, cell culturing techniques are first used. 1955, an enzyme involved in the production of a nucleic acid is isolated. 1956, the fermentation process is perfected. 1960, messenger RNA is discovered. 1961, the genetic cause is understood. 1972, the DNA composition of humans is shown to be 99% similar to that of chimps and gorillas. 1977, genetically engineered bacteria used to make human growth protein. 1978, North Carolina scientists Hutchinson and Edgell proved it is possible to introduce specific mutation at specific sites in a DNA molecule. 1979, the first monoclonal antibodies are synthesized. 1980, the US Supreme Court approves the patenting of genetically engineered life forms. 1980, the US patent for gene cloning is awarded to Boyer and Cohen. 1981, the North Carolina Biotechnology Center is created, the first sponsored research center for biotechnology. 1981, the first genetically engineered plant is reported. 1981, first mice to be successfully cloned. 1982, humulin human insulin drug produced by genetically engineered bacteria, first biotech drug approved by the FDA. 1983, the first artificial chromosome is made. 1983, the first genetic marker for specific inherited disease are found. 1984, the DNA fingerprinting technique is developed. 1984, the first genetically engineered vaccine is developed. 1986, the first biotech-derived interferon drugs for the treatment of cancer are synthesized. 1988, Congress funds the Human Genome Project. 1989, microorganisms are used to clean up the Exxon Valdez oil spill. In 1990, the first federal approved gene therapy treatment is performed successfully. 1992, the structure of HIV-RT is elucidated. 1993, the FDA declared that genetically engineered foods are not inherently dangerous. 1994, the first breast cancer gene is discovered. 1996, scientists cloned identical lambs from early embryonic seed. 1998, scientists cloned three generations of mice from nuclei of adult ovarian cells. 1999, embryonic stem cells are used to regenerate tissues and create disorders that mimic disease. 1990, the Biotechnology Institute is founded by BIO as an independent national education organization. So 1990, the genetic code of human chromosome is deciphered. 2000, a rough draft of the human genome is completed. In 2000, pigs are the next animal cloned by researchers to help produce organ for human transplant. In 2001, the sequence of the human genome is published in Science and Nature. In 2002, scientists complete the sequence of the pathogen Ophrys, a fungus that ruins enough rice to feed 60 million people annually. In 2003, Dolly, the cloned sheep from 1997, is euthanized. This is the history of biotechnology. Thank you.